You will, okay, start from the beginning. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma salli ala sayyidina Muhammad. Okay. Thank you very much to the organizer who invited me to present in your platform. I'm very honored to receive the invitation, and today I'm going to share with you one of our research project. It was conducted with the help of an Indonesian student, where she has attached to our Kuliah of Pharmacy, Faculty of Pharmacy Malaysia, for 3 months. And this project just been completed within 3 months time, which is very excellent student who has helped us. Okay, so this topic today is going to share on the hepatoprotective effect of paracetamol-honey suspension. Next slide, please. Next slide. All right. Okay. Hang on. Sorry. The outline of the previous slide. Sorry. Okay. The outline of topic today is I'm going to show you some introduction and the concept on halal and Thayyiban fusion medicine, which is today the conventional the fusion medicine. We use the paracetamol, which is the conventional drugs, together with the traditional drugs, which is traditional medicine honey, to the in vivo testing on hepatoprotective effect in rats, and some results and discussion. Okay, next slide. Next slide, sir. Next. All right. I think everyone know what is acetaminophen. Previous slide, please. It is also called paracetamol. Okay. So please previous slide. Can we go to previous slide, sir? There are various types of dosage of paracetamol. It is marketed in few type of dosage forms: syrup, suspension, and as well as tablets or caplets. Okay, and not only the single drugs, it appears as single drugs, but also marketed as fixed dose combination drugs with other active ingredients apart from paracetamol. So, for example, you can have when you have an headache, say for example an elderly patient who is on chronic medication for muscle relaxant, and they don't really understood that their own existing medication consists of paracetamol, okay, with muscle relaxant. So when they have headache, they tend to take another paracetamol tablet. So this can easily cause drug overdose of paracetamol. So actually, next slide. Next slide, sir. Okay, here. So basically, if you can see here, I'm sharing with you a few types of drug combination and paracetamol alone. For the paracetamol, we can have syrup, formulated as syrup, suspension, and also tablet, 120 to 150 milligram, etc., etc. And even the tablet paracetamol itself has the dosage strength for children as well as for adults. And then the combination of paracetamol with other drugs like caffeine, it is used for menstrual pain, and like paracetamol plus orphanadrine citrate, it is used as muscle spasm. So basically, easily this PCM is a highly classical drugs, but widely used worldwide. Not only appear as a single API, but also with combination And the market size for this PCM is USD 1 billion annually. Right. So next slide, sir. So basically what we want to tell you here is that paracetamol is very widely used and overdose of paracetamol easily happen. Okay. And the problem with the syrup or liquid formulation in suspension, they usually contain about can contain sucrose or sugar up to 80%. So that will be detrimental to our children here, like shown in the picture, actually my children. Right. They have the reason to add in this sucrose or the sugar in paracetamol syrup formulation is to mask the bitter taste of paracetamol. So, and not only paracetamol, but other sugar or other syrup or liquid formulation, they usually have this high amount of sugar. Okay, this high sugar content in pediatric liquid medications, what we concern is its potential detrimental effect, carcinogenic on that cause dental caries, and this is detrimental to the healthy growing teeth of our children. So what happens if they have frequently have the coughs and colds? For children, their immunity still developing. So this is our concern. Next slide, please. So we inspired by looking at this kind of problem, we formulated an alternative form of paracetamol suspension by substituting the sugar with honey. So it becomes something like synergistic. Okay. So another problem with this paracetamol, when we referring to the other type of dosage form like tablets, okay, the tablet existing marketed paracetamol tablets, it doesn't have any added value, just have give a quick effect on the headache, for example, or as analgesic. Okay, especially for chronic disease, and recently this UK, the NICE UK, which is the National Institute for Health and Care Excellence, NICE, the NIH, National Institute of Health of the UK organization, that usually come up with guidance, okay, clinical guidance for something. So they come up with these painkillers such as paracetamol and ibuprofen. They said should not be prescribed for chronic pain because they do more harm than good. So if we can have some alternative or not only paracetamol but paracetamol with honey, which is the traditionally used medicine, we already know honey is very safe. Honey is has antioxidant effect, has flavonoids content, phenolics, which can give a synergistic or a wholesome benefits of the medicine when we use this paracetamol with honey. So it can also reduce the risk of overdose. In fact, even the acute overdose of PCM can be mitigated if we have an antioxidant administered concomitantly together with this paracetamol dosage form. So risk and the USA. I'm not sure about India scenario, but USA usually they have their established reporting, adverse effect reporting. That's why I could find some information regarding this risk of or the prevalence of overdose of PCM. Next slide, please. Okay, as I mentioned just now, the problems with the existing formulation of paracetamol is the sugar content. Not only my concern or our research team concern, but also other people as far as UK researchers, they also, or the nutritionists of these UK people, they also identify the bad effect of sugar content that can cause their dental concern with commonly used medicines. Okay, so in this example, they are dentists. Can I have my slide? They are dentists and they find out that commonly prescribed medicine for dentists to their patients contain high amount of sugar, and because they're treating patients or children with dental problems, they found out that it is very difficult to cure these problems because and it is related to the high sugar content of the pharmaceutical syrups that they prescribe, which is paracetamol. And not only that, the antibiotics suspension also contain high sugar. Sir, can you give me my slide? My slide doesn't appear on my screen. So the next slide. Hello, sir. Can you show me my slide, please?
Yes, Professor. Can you tell me which slide number do you want?
Okay. After the mail slide number.
Can you please tell us which slide number do you want?
Maybe number 6.
6. Okay.
Number 6 done, and number 7 on sugar content, right? Okay, so next slide number 8. Okay, this table is just to show and share various type of possible possible pharmaceutical syrups, including all the antibiotics type, which the API themselves are very bitter. Similarly to the acetaminophen or the paracetamol, they are the bitter type of API. That's why for formulations in syrup dosage form, we need to add a lot of sugars to mask the bitter taste. So the various sugary pharmaceutical syrups and suspension, and what we found out, the article mentioned that average sugar content is 4 gram per dose. 4 gram is a lot. 1 spoonful equivalent in 5 meal. When we give our children, they has to take 4 gram of sugar. Okay, next slide, sir. On introduction and pain. So not only the sugar content, which is high, but the paracetamol itself can easily can cause drug-induced liver injury, especially when taken by elderly patients. Okay, our elderly patients, they didn't understand, they didn't aware on existing medication. Sometimes they are taking paracetamol with drug combination, and then when they have headache or fever, they tend to just take another tablets of paracetamol alone to cure their fever. Okay, next slide on the next slide number 10, Journal of Hepatology. Okay, this Journal of Hepatology, they also mentioned about drug-induced liver injury, and they found out that acetaminophen can cause hepatotoxicity, which is the most common cause of acute liver failure, ALF, in the US. Even a single overdose can, of the unintentional case, happen, and happening even for the doses that they are taking, the daily doses, recommended daily doses 4 to 10 grams per day. This also has been reported to cause acute liver failure in the US, right? Other issue with this paracetamol is as I mentioned, the concomitant drugs. Okay, factors such as concomitant drugs, systemic illnesses, and some patients, they are also chronic alcoholic abuse patients. So when they take paracetamol with when they are already an alcoholic, their liver already sick, their liver already damaged. Okay, when they take paracetamol, which has the hepatotoxic effect, this can aggravate their symptoms or their hepatotoxic effect. Right. Can we go or move to slide number 11? Okay, slide number 11 is showing example of acetaminophen bottles. Okay, an example of drug associated with intrinsic and versus idiosyncratic DILI, which is the drug-induced liver injury. Okay, there are many type of drugs that can induce liver injury, and particularly we are concerned with the acetaminophen. And for the concept purpose, proof of concept, we are taking acetaminophen as an example in our research because acetaminophen is easily to get. We don't need any special license to purchase, or we are easy to control in our inventory. So that's why we are taking acetaminophen as one of example to show that when we combine with traditional drugs such as honey, we can reduce or minimize the effect, the bad effect of this drug on our organ. So today on our liver. Okay, can we move to next slide, sir? Slide number 12. Okay. All right, slide. This slide is showing WHO Sustainable Development Goals. Okay, I'm sure everywhere in this world we are aware about these WHO Sustainable Development Goals. There are 17 of them, and what related the one related to our research area, our academician profile is number 3 on the good health and well-being, number 9 some innovation, and number 16 partnerships for the goal. Okay, partnerships for the goal actually meant not only between the academician with the industry players, but also we are together with all the publics. If the publics can listen and we can increase the awareness on this fusion medicine, the conventional together with the traditional medicine, which can have a synergistic effect, this would be great. Okay. And this because IKOP is we pronounce as IKOP. Okay, it is we are our vision is to produce halal and thayyiban medicine, but because especially on research base, we are targeting this fusion medicine, the halal parts and the thayyiban parts, but focus on the fusion medicine, not on the single API conventional, but we are targeting to produce a better medicine, which is fusion medicine, which to us, it should have this kind of attributes, WISE, wise. It should have a wholesomeness attributes, medicine that care and cure and actually treating the disease. It should have an I aspect, innovation aspect of medicine. It should also have the S, sustainable and synergistic effect. Sustainable, which means the medicine is easy to get. The medicine also is actually eco-friendly, okay, and it produces synergistic effect when combined together. And E, it should be equitable, basically referring to the price. Later on, the price will not be so expensive. It should be accessible by anyone, whether they are farmers or they are lawyers. These are kind of wise medicine we are looking forward to produce in the future. So these are the characteristics of the fusion medicine. Halalan Toiban, fusion medicine, we are in our mind, in our vision to produce. Okay, sir, next slide, please, sir. Slide number 13. Okay, this slide showing our artwork of the products. Paracetamol, honey suspension, and we are using paracetamol, the real raw material paracetamol powders, not usually sometimes when we do research, we tend to buy tablets of paracetamol, we crush and we mix together with honey. But we are not doing that. We are buying from the original raw material supplier of paracetamol in the powder form, and then we mix together with honey and some excipients to make it stable. Okay, so this is our formulation. Okay, next slide, sir. Okay, this the paracetamol is very conventional drugs. The approved indication is as antipyretic and analgesic. For our formulation, we actually already done an animal study to compare the synergistic effect with paracetamol alone. Okay, when we compare, the synergistic effect is being demonstrated with our fusion medicine. We got drugs, a medicine that can reduce the body temperature, better and prolong reduction in body temperature, as well as the analgesic effect, prolonged anti-pain effect when we tested with animals. Okay, but today I'm going to share with you the hepatoprotective effect of this paracetamol honey suspension. Okay, next slide, sir. Okay, honey. Honey itself is traditionally, I think all of us know what is honey. Can be various type of honey can be produced depending on different type of flowers. The bees take the nectar of that flowers. So major components, honey actually also available in pharmacopoeia. The USP, BP, they mention about the specification of honey. Okay, so for honey, it has major components as sucrose, fructose and glucose, and it has low glycemic index, traditionally used to reduce fever and demonstrate hepatoprotective activity, antibacterial effects, and anti-inflammatory effects. So we have chosen honey to be the traditional or prophetic medicine to be fused and formulated together with the conventional drugs, which is paracetamol. Okay, next slide, sir. All right. So for today, I'm going to share the hepatoprotective testing that we have conducted in rats. Okay, we use spread dolly albino rats, 24 healthy males, and they are aged 5 to 6 weeks, And we conducted this after we got the approval, ethical approval from our IIUM, International Islamic University Malaysia Ethics Committee. So next slide, sir. Alright, after we got these rats, we acclimatized the rats for 7 days with normal feed and tap water. Okay, and then we divide them into 4 groups. Group number 1, each of the group consists of 6 rats, and group number 1 doesn't receive anything. They just receive all normal tap water and normal feed and tap water. Okay, group number 2, they will be receiving, they receive the paracetamol with sugar suspension. Group number T2, we call it T2, is going to be treated with paracetamol honey, and the final one is with honey alone. So altogether we have 4 groups. Number 1 is control, is normal, doesn't receive any intervention or any paracetamol, not honey at all. Okay. Number 2, receive paracetamol suspension sugar one, which is the commercially available paracetamol sugar. Okay. Then we compare with the paracetamol honey, the one that we make, and honey alone. Okay. After 7 days, during the day 8, we give them 2 grams of. Before treatment, we take some blood samples. Okay, just to make sure all of them have a healthy biomarkers that we are targeting, which is the AST and ALT. Okay, AST and ALT is the liver enzymes, which are very important biomarker for identifying whether our liver is healthy or not. Alright. So when we before the day of treating, we take their blood samples, and then after the next day, we give them the paracetamol with sugar, paracetamol with honey, and honey alone. Right? After 24 hours, the dose that we gave them is actually a toxic dose. A dose that from established literature, this dose 2 gram per kilogram body weight can give hepatotoxic effect on the rats with single dose administration. Okay, after 24 hours, we euthanize these animals, and we collect the blood. Okay, we have to sacrifice the rats to collect the liver. And the liver, we do the histopathology examination as well. Histopathology examination, yes. And that the blood we send out to check the level of ALT and AST, comparing the blood profile before paracetamol intoxication and post 24 hours intoxication. Okay, next slide, sir. Slide number 18. Alright, this is just to show you the example of acclimatization. We have the rats put in the cage with normal feeding and tap water. Okay, on the right hand side is where we show or demonstrate the force feeding technique to the animals. Okay, next slide, sir. Slide number 19. Alright, these rats already euthanized, already anesthetized. When we want to collect the blood before the treatment day by cardiac puncture. Okay, can we move to the slide number 20? After the treatment, we have to, after 24 hours, we have to sacrifice the rats and take out or remove the liver for further histopathological examination. Okay, can we go to slide number 21 on the results and discussion? Alright, here the graph on your left is showing the rats food intake from day 1 until day 10. Day 10 where we sacrifice them. Okay, as you can see, during day 1 until day 7, during the acclimatization day, they are very healthy. They take food regularly, and when we start to do the cardiac puncture and take their blood before a day before treatment day, they start to they suffer from stress, a bit of stress, and that's why the food intake reducing on day 8 until the day 9 where we where we give them by force feeding the paracetamol suspension, and day 10 we sacrifice. Okay, so on the right is on body weight. Body weight doesn't really show any effect even after cardiac puncture. Okay, but from day 1, of course, after they taking several days after taking food, they start to gain weight. Okay, can we go to next slide, please? Slide number 22. Okay, after intoxication on day 8, we sacrifice them 24 hours after the single dose of toxic dose paracetamol. Okay, then we take the blood and we measure the ALT and AST. Okay, ALT and AST. Oh, sorry. Okay, this figure actually showing you the ALT pre and post. Okay, the similar diagram we obtain for AST as well. So for the ALT liver enzyme, as you can see here, pre-treatment, the blue graph, the blue bars, all of them are not statistically significant. All same alphabet. Okay, so basically during before treatment, they are all very fairly healthy and have same ALT level. Okay, after the treatment day on day 8, when we take their blood and measure the ALT, we can see here this this ALT level for paracetamol sugar suspension becomes tremendously increased. Okay, PSS is the paracetamol with sugar formulation, which is the normal formulation. At 2 gram per kilogram body weight, their liver damage. Okay, the hepatotoxic happen with this normal sugar paracetamol formulation. In contrast to in contrast, the formulation that we formulate with honey at same strength, 250 milligram per 5 ml of paracetamol suspension, but we add in honey, substitute all the sugars with honey. Okay, about 70%, 60 to 70% of honey is present in the formulation. So it shows that this presence of honey can greatly reduce all the toxic effect of the liver. Okay, PHS here um, is the honey, paracetamol honey. Okay, when we compare with honey alone, yeah, alhamdulillah, honey alone of course doesn't cause any hepatotoxic effect on the liver. It's very safe, right? Okay, this is ALT on the left, and on the right is AST. Similar trend, ALT and AST. They are the groups, the animal groups with that is intoxicated with paracetamol sugar suspension. Their liver profile become very bad. Their AST and ALT increase. Okay. All right. Can we go to slide number 23 on liver histopathology? Okay, this liver of the animals of the rats shows that we have 4 liver sectioning here. The first one A is the liver from the control group, which doesn't receive any honey, doesn't receive any paracetamol. Okay, they show a very healthy liver cells, right? On the B, picture number B shows the liver, damaged liver, liver from the groups that receive paracetamol sugar suspension, the normal one in the market. At the 2 gram dose, single dose, their liver become distorted, and the architecture is not normal. The CV, which is the central vein, distorted as well, and this indicate their liver injury of the rats that receive paracetamol with sugar suspension. Okay, the liver of the rats that receive paracetamol with honey, our formulation, they show a little bit of toxicity effect, but their CV, which is the central vein, is protected. Okay, they are also almost near to normal healthy liver. Okay, the liver of honey groups also normal and very similar to the group of rats that doesn't receive anything. Okay, so basically here from the liver histopathology data, we can safely mention that the presence of honey in paracetamol suspension can protect or reduce the toxic effect of paracetamol. At 2 gram dose, which is the toxic dose of paracetamol, presence of honey able to minimize the toxic effect. All right. Can we go to the next slide, please? Slide number 24. Okay, this slide number 24 is to show you the paracetamol mechanism of toxicity. Okay, basically paracetamol, when we take paracetamol, it will be converted to NAPQI, and when at toxic dose, our own glutathione will be depleted. They cannot convert and cannot render it safe, but it will so complex pathway here. It will finally cause cellular death. Okay, so paracetamol poisoning is happening. Sometimes in hospital we have not sometimes, in the hospital itself we have paracetamol blood monitoring. Whenever there is adverse effect of patients being admitted, even the pharmacist has to quickly come to the hospital, even at night, to settle this issue. Okay. All right, so can we go to the next slide, 25? I have 3 more slides. All right, slide 25, results and discussion. Right, so basically the paracetamol intoxication, as you can see, at 2 gram dose can cause remarkable liver damage. Okay, indicated by the reduction in the, perhaps due to the oxygen, reduced oxygen perfusion. So this causes the development of liver diseases, and which progress to the cell death. And as you can see from the histopathological data, it is characterized by congested central vein, dilated sinusoidal filters, and fatty changes, and loss of architecture of the liver. Okay, as mentioned, it is due to the NAPQI free radicals resulting from the excess metabolism of paracetamol, which interferes with protein synthesis and production in the ribosome. As the results, this kind of proteins and lipoproteins, they cannot enter out of the plasma. They form the triglycerides, and stay there and cause the fatty accumulate in the liver cells. Okay, slide number 26, second last slide. Right, honey treated architecture and less extent of hepatocellular hypertrophy, which confirm the hepatoprotective effect of honey. And based on the liver histology of paracetamol honey suspension, the presence of honey in our formulation show protection of honey is there. And when we compare with our results of phytochemicals results, that quantify the phenolic and flavonoid content of our honey, it shows a good corresponding data. It corresponds with the increase in phenolics and flavonoids paracetamol as compared to paracetamol normal sugar formulation. Paracetamol sugar formulation, they doesn't have this phenolic and flavonoids. But paracetamol honey, yes, we do have this phenolic and flavonoids level, and which this can eliminate the radicals, free radicals, it can inhibit oxidative damage and consequent degeneration of the liver tissues. And then in the honey itself, there appear some of the honey has non-protein sulfur-hydryl groups, and because some of the honeys of they may contain amino acids and other micro elements. Okay, and this may have a role in the prevention of toxin-induced morphological changes, including the central vein distortion or degeneration in the liver. Okay, so the results, what we obtain here, histopathological results and biochemical level, determining the AST and ALT level, it shows that our the result is corresponding to each other. High level of phenolics and flavonoids give a protection of the toxicity of paracetamol, protection at the liver. Okay, last slide, sir. Conclusion. Right. I think everyone know now that paracetamol can easily be, our anyone can easily get overdose of paracetamol, but the susceptible group is the children and the elderly patients. Okay, risk of overdoses of paracetamol, however, can be greatly reduced if we can concomitantly administer something antioxidant which is very safe, like honey. Okay, but the taking of paracetamol separately with honey, it will cause some lower compliance and will increase the cost of to buy the honey, cost to buy the medicines. So what we are targeting here, we want to reduce the cost, we want to increase patient compliance. That's why we do the fusion medicines. Okay. So the results of this study demonstrated the hepatoprotective action of honey in paracetamol honey suspension that we formulated. And even at the toxic dose that with presence of honey can reduce the animals. So what if we just use the normal dose of paracetamol? So inshallah our liver will be protected well, especially the liver of the elderly patients, which is already worked very hard over the years, especially those patient elderly that has to take polypharmacy. They have so many drugs to take. Their liver already worked very hard to metabolize all these drugs. So this kind of protection is should be in place to help them and to protect or to prevent further damage. Okay, the protective effect of this honey may be due to their antioxidant property. And therefore, this paracetamol honey suspension can serve as the newly introduced analgesic and antipyretic drug that have a hepatoprotective effect. Okay, the analgesic or the pain, even the chronic pain, these are very common pain. Most people, I think, maybe 90% of us has experienced pain. Okay, and has experienced headache and fever. So we hope that with this kind of concept, fusion medicine, utilizing conventional drugs or utilizing contemporary drugs, API, we fuse with the established traditional drugs like honey, can give a synergistic effect and can reduce the adverse effect of the existing drugs. And this similar fusion medicine, I think we should encourage and we should advocate the use so that many more studies will be conducted by anyone so it can prosper. And the partnership with the SDG goal number 17 that I mentioned earlier, to achieve the goal with partnership, we need to partner with academicians, need to partner with the industry, and both of us need to do proper education to the public, so everyone are